All right, afternoon, everybody. Good to see you. Thanks for coming out. Today is ATP, cellular respiration, and photosynthesis. I'm supposed to be at the softball fields at 5, so we're going to quit at 4:50. And yeah, all right, we'll get rolling. We'll do a little bit of notes type stuff, and then towards the end or throughout, actually probably between some of these different subjects, we'll do some practice free response. Some of the free responses have changed over the years, so I'll kind of tell you how similar it is to now or how different it is, stuff like that. And then when I see you guys in class, that's when I'll tell you the format of the test and all the basic things like, hey, he never told us how many free response there are or how long they are or how much time we get or anything like that. I'll do that in class. If any of y'all want a lab stool, the lab across the hall that we usually use for our labs is open, so you're welcome to go and grab one if you like. And other than that, let's get rolling. So the way that this works, if this is the first one of these that you've been to, is it's mainly just like you sort of participating in your own head or answering out loud, but because we have so many people, the main idea is just that you're checking to see if you know this stuff. So one of the things that helped me a lot in college, especially, I thought I kind of had a handle on it in high school, but then in college it's sort of next level, is knowing what I knew and knowing what I didn't know. So when I have a test coming up, it's here's all the crap that I know, here's the crap that I don't know that I actually need to study, if that makes sense. So figuring out what I should spend my time on. All right, so take maybe 30, 45 seconds and jot down everything you know about this. You might start with, what is it? If you know what it is, then tell me what makes it special and all that kind of stuff. Here you go. Busy. Here you go. Thank you. Yo. All right, time's up. So what is this molecule called? Yeah, ATP. And what's the TP stand for in ATP? Triphosphate. Yeah, it's adenosine triphosphate. So what's interesting about going back and reviewing stuff from the beginning of the year is now that you know more stuff, you kind of look at things differently. Like when we first learned about ATP, we didn't know we hadn't done genetics yet. But now that we've done genetics, that's an adenine. What's that? And you might know in general or you might know specifically what it is. What do I know about that, even if it's pretty general? Do what? Very close. It's ribose. So it's a type of carbohydrate. Specifically, it's ribose. If it was deoxyribose, it'd be missing that oxygen right there. We deoxy it. So that's an adenine. That's a ribose, and that's a phosphate. So what is a adenine, a ribose, and a phosphate? Isn't that an RNA nucleotide? Don't I build RNA out of a phosphate, a sugar, and a base? And if that sugar is ribose, then this is an RNA nucleotide. If that was deoxyribose, that'd be a DNA nucleotide. So if I take a piece of RNA, that's a legit RNA nucleotide, not it's similar, everything that's not covered up right there is an RNA nucleotide. If I wanted to build RNA, then if I tack on a couple of more phosphates, now I have ATP. And so this goes back to the whole idea of anytime you have a molecule, if you tweak it a little bit, that's probably another molecule that does something. And that relates to evolution, because if I have a molecule, and let's say I'm trying to build it, and I build it slightly incorrectly, then now that molecule probably has a new function. Think of the most recent unit. I have cholesterol. All of a sudden I tweak it a little bit. Oh, now it's not cholesterol, it's ruined. Oh, no, it's not, it's testosterone now. Or, okay, well, now I take testosterone. Change this little chemical. Oh, you ruined it. No, I didn't. Now it's called estrogen, and that does something. So most of the time that you look at all these molecules and stuff, when I go and I start kind of like looking at stuff and you're kind of like, hey, how could I, with evolution, how could I go from this molecule over here to this molecule? Like there's like 8 steps in between and every single step does something. So if I start looking at anything up here, here's some chemical, it does something. If I tweak it just a little bit, I put an OH at the end, that's a different molecule and it does something. If I tweak it just a little bit, it's a different molecule and it does something. If I tweak it, that does something. So that's kind of how some of this stuff fits together. What was so special about ATP? Yeah, so it's kind of the main energy source for everything. All of these chemical reactions, I won't zoom in again, but all these chemical reactions finally end at the very bottom. Actually, I guess I will zoom in with ATP. So the reason ATP is important, the reason it's one of the few molecules out of all this crap up here that we have to be able to recognize is like all of these arrows feed into eventually making ATP. And that's the spinning ATP synthase right there. So why was it that ATP was the energy molecule? Like what made it special compared to all the other crap in the world? Yeah, it's highly unstable. It breaks apart really easily, and at first that sounds like a detriment. Which part makes it highly unstable? Like where on there is it unstable at? Yeah, the phosphates. And what about the phosphates? Yeah, they're negative. So I have negatively charged phosphates next to negatively charged phosphates next to negatively charged phosphates. This was like one of the original times. Actually, this is the second unit where like phosphate being negative came up. But after like unit 2, doesn't phosphate being negative come up in like every single unit except like ecology maybe? Seems like that always comes up. In lab, it's coming up all the time. So those phosphates are negative. So those guys are like always repelling each other. So because of that, if I like snap one of those bonds between one of these phosphates, it releases a ton of energy. You might remember, I hit record, so we're good. You might remember in the enzyme unit, we had a reaction like that. What kind of reaction is that if it starts high and ends low? Exothermic. What was that bump called? Activation energy, and that's how much energy I have to input in the reaction to make it go. Over here, if I want to do a chemical reaction of like breaking off a phosphate, I have to input it some energy to break that bond. Do I have to input a lot of energy to break that bond? No, why not? It's unstable because they're already repelling each other apart. Make sure if you're writing for a professor or for the AP Bio test that you'll be like, oh, it's unstable, but then tell me like why it's unstable. Don't just say like it's unstable. They're like, well, how do you know it's unstable? Like here's some other molecule. How do you know that it's more stable? Oh, well, these negative things repel the negative things. That's why it's unstable. So if I input a tiny amount of energy right there, it releases a ton as they go flying apart, essentially. And so my chemical reaction looks like this. On a technicality, this will bother some of you guys. So if it gets confusing, just let it fly. Technically, it's not releasing its energy until this phosphate like bonds with something else. So this, again, if it makes sense, cool. If not, blow it off. Just trying to be technically correct since I'm on a video. Let's say you're in our current unit that the one that we just tested on. I've got a protein that's got to activate a protein, that's got to activate a protein, that's got to activate a protein. That's a transduction cascade. And so what I'll do is I'll have this protein, and usually what it does is it adds or removes phosphates to things. So it'll normally break down an ATP, add a phosphate, and that's what like turns this on. So whenever we're always talking about proteins, turning on proteins, one of the common ways to turn on a protein is to peel off a phosphate from an ATP over here, add it on, and now this guy is active. He will then go and grab this protein, take a phosphate from an ATP and like added on there. So we call that phosphorylation. We're phosphorylating these proteins. So a lot of times whenever you activate a protein or turn it on, you're adding a phosphate. If you're in a chemistry class, they'll basically say, like, you aren't releasing energy by breaking off this phosphate. You're releasing energy when you break this phosphate and it, like, adds to something else. I'll tell you why just once, and if it doesn't make sense, don't worry about it. Let's say that this has an energy of 20, and if it's bonded to this thing over here, it has an energy of 10. That's how I know it gave off energy. This has an energy of 20. This has an energy of 10 that it just made, so it gave off 10. But a chemistry teacher would say, when you break this off, you haven't given off energy until it bonds with this, like, lower energy thing. All right, just technicality. Okay, so now let's try, before we get into photosynthesis and cellular respiration, let's take a crack. Which one do I want to do first? 2019. All right, so back in 2019, they asked this question. This was a 4-point question. You guys get some 4-point questions on your test. You'll have 2 questions at the beginning that are going to average 9 each. So question 1 and 2 together are going to be 18 points. Might be 8, might be 10, but together they'll be 18. And they're going to have 4 4-point questions. So this is what the 4-point questions kind of look like. Nowadays, they're a little bit more wordy. So let's give this one a crack. Let me switch cameras and I'll read it to you because I realize some of you guys are in the way back. All right, so it says the pyruvate dehydrogenase complex. You're like, dang it. Catalyzes the conversion of pyruvate to acetyl-CoA, a substrate of the Krebs cycle. You're like, Krebs cycle? I recognize that one. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. Based on the way you're reading this, are they expecting that I know much about pyruvate dehydrogenase complex? Are they expecting that I've ever heard of this disorder? No, so they expecting me to, like, they know that I'm seeing this for the first time, but maybe some of this stuff I am supposed to know, but, like, they are giving me some new information. Okay, so A, identify the cellular location where PDC is most active. B, make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle in a cell. That'll be a point. I don't know if you can tell, but that's in bold. Make a claim is bold, so that's a point. Provide reasoning is bold, so that'll be a point. To support your claim based on the position of PDC catalyzed reactions in the sequence of the cellular respiration pathway, and I won't even read this one yet. So a point for that, a point for make a claim, a point for provide reasoning, and then a point for part C. I picked this question on purpose because I don't like it at all. But you will come across questions on the AP Bio test. Every year there's one on there that I'm like, this little part in there is unfair. So give it a shot. Jot down what you think. You don't have to write in complete sentences for me. Just kind of a gut check. Who needs to sign this? Is it missed like any major group? All right. There you go. You're welcome. If you guys would rather be up here, you can. It's totally up to you. Or if you want, you can grab a stool from across the hall. Either way. All right, so take a moment, see if you can jot down A or B. If you're timing yourself, you're supposed to be able to do this whole question in 7.5 minutes. Got it. I get you. Thanks. You're welcome. You're welcome to grab a stool, or if it seems weird, like, you're welcome to, like, sit right along here, or if one of you guys want to take the captain's chair right there, you can.
I'm sorry.
Okay. It is. My wife and my son bought me that as a surprise one year. My wife and my son. I'm sure that he just helped wheel it up here. I'm sure she paid for it. All right. So, to answer this, here's one of the things that we have to kind of know. But here's the part that I don't like. Part A, I don't like. They have a very clear answer. We can even go and look at the rubric. By now, you guys probably trust me. So if I just say like, oh, the rubric says this, or I think this, but the rubric says this, certainly if it was at the beginning of the year, I'd prove everything by showing you the rubric. But for part A, so here's what happens. If I'm inside of a cell, what was this process that happens outside the mitochondria called that's glucose to pyruvate? Glycolysis. And why was that a logical name? It was glycolysis. What is glyco? Sugar, or specifically glucose. And then what is lysis? Yeah, breakdown. So this means sugar splitting, and so we do glycolysis. So I start with one glucose. We don't have to know all of them, but we technically go through 10 chemical reactions to get here. So this is like a summary of like 10 chemical reactions. And I end with like 2 pyruvates. And along the way, does anybody remember there were like 2 main things that we looked at that like along this arrow that I don't currently have up there that I need to add? Yeah, I think I heard NADH, and what was the other? And ATP. So I do end up like I input 2 ATP, and I get to output 4. So overall, I'm like plus 2 ATP. I made a profit of 2 ATP. Kind of like I spent $2, but I made $4, so I made $2. Yay. Then I also come in and I have some NAD plus that becomes NADH. And it's going to go inside my mitochondria. And I'll try to kind of move around. That way I'm not like blocking the same people over and over again. So if you're like, why does he keep doing this? That's why. All right, so I do that. What happens to the pyruvate? Does it just stay there? Or? Yeah, it goes into the inside of the mitochondria, becomes acetyl coenzyme A, acetyl-CoA, and then what was that cycle that it's now a part of? Yeah, the Krebs cycle. So some professors call it the citric acid cycle. Most recently on the AP Bio test, they've kind of used both names, but in the past they would pick one or the other and stick with it the whole time. So if you're writing an essay and you call it Krebs, you're good. If you're writing an essay and you call it citric acid, you're good. But occasionally they'll just pick one, and like on the multiple choice, they just stick with that one, so you need to be able to recognize both. So be able to pull one of them out of the air, but be able to recognize both. So then I go through like the Krebs cycle. Krebs cycle. All right, I had 4 things that kicked off of the Krebs cycle, and we will come back to this in just a second. Does anybody remember I had 4 things? I would kind of draw arrows about like this in class probably. Actually, this one didn't go like that. I probably had those just go like that. There were 2 main ones that I cared a lot about, and then 2 other ones that were kind of meh. Yep, CO2. So I kick off some carbon monoxide. Yeah, NADH, good. So I also produce some NADH. Very close. So well, that's true. So coming in here is NAD plus, and it's going to become NADH. That's true. What's that one? So the most important two are like these arrows. Yeah. So FAD comes in and becomes FADH2. All right. Lastly, I get like a measly 1 ATP. Okay. What was important about the NADH and FADH2s? Like, why are those the most important ones? What was their job? They have the same job. Yeah. So they're going to feed this chain. What was this chain of 3 pumps right here called? The electron transport chain. So where are they going to feed it? Electrons. So these guys are feeding electrons, to these pumps. Oh, they quieted themselves down. Nice. So yeah, if you need to, feel free to shut it. Yeah. So those are going to feed electrons to this. All right, we're going to come back to that in just a second. So where does PDC do its job? What do you think? What does it start? What does PDC start with? Like, what's the reactant for PDC? For pyruvate dehydrogenase complex. It's an enzyme, so it runs a reaction. So it'd be like PDC. What's on the reactant side and what's on the product side? What? Yeah, pyruvate. It's going to convert pyruvate into acetyl-CoA. So over here on the reactant side is pyruvate. Over here is acetyl-CoA, acetyl coenzyme A. By the way, now that it's further along in the year, have we heard acetyl again in a later on unit? Where? Or what? What was it doing? Who got acetylated? Yeah, you're in the right unit, genes. What got acetylated? No, promoters got methylated. Histones. I don't know who said it. There you go. So we can acetylate histones to turn genes on and off. It's amazing how much crap you've forgotten in this class, isn't it? Like how much you've known and how much you've forgotten. You know why? History, government, it's taking up room in your brain. So get your mind right, dump that crap, replace it with biology. So where would it be active? You're like, well, I'm not sure. Maybe it's out here, maybe it's in here. It happens to be in here. And they only took if you said in here. So identify the cellular location where PDC is most active. They would say inside the mitochondria. What's this area kind of just out here in the open part of the mitochondria called? The matrix. So they wanted the matrix. Now, if I am a AP Bio student, I'm going to have a hard time determining how I know that it's not here, because I don't really see. All books show pyruvate there. All of them show acetyl coenzyme A there. They don't all show all the reactions in the middle. So I'd have a tough time knowing which was which. But sometimes you get screwed. That's just how it is. Yeah. Well, that's why I don't like this question. Like I agree with you. Like for me, I knew that, but I'm a teacher. So I wouldn't expect a college level AP biology student to know that. But last year, one student out of like 300,000 got a perfect score on the AP Bio test. Because of crap like this. Like, how do you get everything right, not make any dumb mistakes, and then like get some of these random trivia things right too? So I'm doing this on purpose because when you take the real test, you will come up against a question like this. You're going to come back here and I'll be like, how was it? You'll be like, this is fine, this is fine, this is fine. What the hell is going on with this little parasite, whatever? And you're like, I don't know, I'll have to see the question. So there'll be unreasonable things. So the reason I'm telling you this is I'm supposed to knock out this question in 7.5 minutes. So if I know my stuff as well as the best AP Bio students, you'd be like somewhere between here and there. I don't know, cytoplasm or matrix. Can't think of a good reason. I'm going to give myself 10 more seconds. Then I'm just going to guess and move on. You can't sit there and stare at your paper for like 7 minutes. You will die. So finishing the test on time is hard. However, you guys have an advantage. We're already done. Most people on the AP Biology like group things that I'm a part of, most of them are still back in genetics, or they haven't started ecology or evolution, and a lot of them don't review. So all the stuff you're feeling now, they're going to feel like on test day. So you might be like, this sucks, but it sucks for all the students. So you'll be fine. You've got an advantage. Wait, say that again? So a point is a point is a point. So like, let's say that this one right here is worth 1 point. Let's say the next one was what's 4 plus 4? It's 8, and that's a point. They're both equal. Yeah. They do weight the free response and the multiple choice slightly differently so that they're even. So a point on free response is worth a little bit more than a point on multiple choice. So, but I'll explain that kind of part in class. Okay, make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle in a cell. So take a moment. See what you would think. All right, so here's what they accepted. I have to get both of these right to get this point, because I have to make a claim about how it affects glycolysis and how it affects the Krebs cycle and NADH production. So normally I'd be like, hey, what do you guys think? But for here, since PDC is acting here, here's what they say. I also slightly disagree with this. This is the question I dislike the most out of like the last decade. Most tests, I'll find one thing for 1 point on all the free response for like that year where I'm like, that was kind of mean, but whatever. This one, I've got two things that I kind of don't like, and it's in the same question. So what they would say, and as a teacher, if I'm answering it, I know what they want me to say, but I still don't like it, is because PDC acts right here, it has no impact on NADH production in glycolysis. But it has a major impact on the Krebs cycle, right? Like, if I'm going from here to here, I'm making an NADH before I get to this part. So if this part's messed up, this part's still running fine. If this part's messed up, then none of this is happening. Does that make sense? So what they're wanting you to say is NADH production and glycolysis is largely the same or doesn't change much, or however you want to say that. Whereas in Krebs cycle, they want something negative, like it goes to 0, it goes down, or it's less. If you said anything of like going negative, you're right. What I don't like about that is if this backs up, then it probably backs up all the way up here also. So you might argue that pyruvate could start taking an alternate pathway like fermentation, but like, again, for an AP Bio student, I'd be like, but if it backs up here, no matter where it backs up, it would back up all the way. They were wanting you to think, hey, if I have a ton of pyruvate, then maybe I'll just start like converting it into like lactic acid. So I picked this one because this question is kind of brutal. All right. The last one, PDC deficiency is caused by mutations in the PDH1 gene, which is located on the X chromosome. A male with PDC deficiency and a homozygous female with no family history of PDC deficiency have a male offspring. Calculate the probability that the male offspring will have PDC deficiency. Anytime you're doing Punnett squares, if it's on a free response, you can answer in decimals if you like that. You can answer in fractions. I'm a fractions guy. I should probably work this. All right, give you 30 more seconds. Sorry if I'm looking over you. I'm just switching. No, you're fine. I'm just switching the cameras there. All right, what are the odds? 0. I can't. So the reason I know that is it says this. It's caused by a mutation in this gene. It's located on the X chromosome, so the Y doesn't carry it. That makes sense. Pretty much all sex-linked traits are carried on an X chromosome, because otherwise, if it was carried on a Y, then like females would never have the gene. A male with PDC deficiency. So I've got a male, and he's got it. I have no idea if this is like dominant or recessive. So I'm just going to put like bad. So you know how a lot of times we'll put like big B or little b? I'm just going to put like bad. So he's got the bad gene. We got a homozygous female with no family history of it. So it doesn't say that she's got the disease. It says that her family, it's not in her family history. So her X's must be fine. So when they have a boy, the dad is going to cough up a Y chromosome. So I'm definitely getting a Y from dad. And then that means I have to get my X's from mom. So I'll be fine. If they had a daughter, then they're going to get, bless you. If they have a daughter, then they are going to get, I'll just use capital B for the gene. I have no idea if it's dominant or recessive. So if it's dominant and the daughter's got it, they'd be screwed. If it was recessive and the daughter's got that copy from dad, they wouldn't be. But they asked me about the sons. This is what can make the AP Bio test hard. You get 90 minutes of multiple choice. You get a 10-minute break. You go and you like guzzle a Coca-Cola and eat a Snickers. And then you come back and you go through, you're halfway through the free response and you get to this. And your brain's tired, but you have to go fast. Now, I picked one of the meanest ones, one of the toughest ones for the best shock value. So if you're like, oh my gosh, I feel totally unprepared. Well, yeah, you're pretty unprepared for this question. So, but that's as expected. So they aren't all like that. Some of them, it comes down to stuff to where you're like, makes ATP? Powerhouse of the cell? Not exactly, but some of them will seem easy. So let's go and look at this. What is that? ATP synthase. First of all, what is this organelle? Mitochondria, and what was that? ATP synthase. What am I getting ready to ask you about ATP synthase? Yeah, I'm going to ask you two things that are important about it. It's hollow and it spins. Why do I care that it's hollow? Yeah, what is it that passes through? Protons. When protons pass through from high to low concentration, exactly, that is what causes ATP synthase to spin. So as it spins, what is it accomplishing? Like, why do I give a crap that this thing spins? Yeah, what am I slamming together? See ya, good luck. So I can be taken a lot of different ways. Yeah, an ADP and a phosphate, and what does that make? ATP. So I have adenosine diphosphate, ADP, and a phosphate. Smash those together, ATP. So if I can make that thing spin, I can make ATP. How do I make it spin? Protons flow through it. I want to make sure that protons always flow from this space to this space. Now, you guys already told me a moment ago that this space out here was called the matrix. What was this space in here called? Yeah, the intermembrane space. Intermembrane space. Inter means between. Membrane and space, that's obvious, but it's the space between that membrane, the inner membrane, and the outer membrane. That inner membrane was all folded. Why did I care that it's folded? Yeah, more surface area. What's on the surface that I want? Like, I don't care about the surface area on the outside. Why do I care about the surface area on the inside? Yeah, more ATP synthases and more black boxes. What are those black boxes? Yeah, pumps. That's how I always draw pumps. My pumps are always black boxes. Even if you're looking at neurons, anytime I draw a pump, it's always a black box. What kind of pump were these? Proton pumps. Now, what powers them, though? Electrons. So they're proton pumps. Huh? Yes. The question was, in all cases, do pumps always move stuff from low to high? Yes. Because if something was going to go from high to low, we could just have a channel and stuff would flow from high to low for free. So pumps cost energy. So we only use a pump if we have to, and that means we're going from low to high. So yeah, there's no, out of pure, I hardly ever say this, but like based on definition, they have to always move stuff from low to high. Yeah, it wasn't just by chance. Yeah, it wasn't just with the neurons going against. It's also here going against, so pumps will always go against because pumps are always active transport, so they always require energy. And then I know that we said it, but what was the energy that powers those pumps? Electrons. We don't really want to use ATP to power them because this whole process is trying to make ATP, so we'd like to not use a lot of ATP in our ATP making process, so we use electrons. Remind me again really quick, how could electrons carry energy? Like, how do I? Yeah, they drop in, what were those levels called? Energy levels. So again, try to always be specific. If you say something like, and watch out for pronouns. You might even have other AP teachers that like give you crap about pronouns. So be careful of when you're like talking about this and you're like, they provide power for them so that they can pump them inside of it. And you're like, I don't know what you just said. Like, they provide power, meaning electrons, to them, meaning the pumps, which pump them, meaning protons, into it, the intermembrane space. Like, I don't know what you're talking about. So be careful of whenever you say it and they and them in a biology essay, unless it's very clear, like, what do you mean it? But a lot of times students will say, like, it gives energy to it so that it can run it. And you're like, I don't know what you're talking about. She told her this, so then she turned around and hit her after she said this to her, and you're like, who got punched? Like, I don't know what's going on. So intermembrane space and matrix. So if protons flow through there from the intermembrane space out into the matrix, that thing will spin and make ATP. That means proton pumps want to push them back. Kind of like in the nervous system. Wasn't the whole point of the sodium potassium pump to basically just like reset everything back like it was? Push the sodium back out, push the potassium back in so that we can go again. I want to keep like, you know, doing this. So I have to just sort of like push everything back to where it was. You're starting to see like, even though a lot of you guys knew this really well back then, you're starting to see themes of like, holy crap, like pumps seem to always just reset stuff. That's because pumps usually just reset stuff. All right, I needed electrons to power those. And as you guys were just saying, when electrons fall in energy levels, they release energy. Who are my electron carriers again? Who are my electron carriers? Not the ones that run on, not the pumps, not the proton pumps, the electron transport chain, but who carried the electrons there? Yeah, NAD and FAD. On the AP Bio test, they typically just put those in combo. A lot of times it'll just say something like this, like NAD plus / NADH. Like, what's the job of this? And those are the same molecule. It's just when I'm empty-handed, I'm called NAD+. Once I've grabbed my electrons and I'm full of electrons, I'm called NADH. For me, if you gave me these letters, it's logical to me because I think of like NAD+, like NADH has more letters, so it's like full, so that's the one that's like holding electrons. In my brain, that makes sense. This one is sort of empty. Now it's full of electrons because now it's got like more letters. Again, in my brain, this makes sense. Not saying it makes sense in all brains. All right, so the Krebs cycle. What was the point of the Krebs cycle then? It's just kind of spinning around in a circle, it looks like. What is it doing? Looks like it's kind of accomplishing nothing. That one's no good. All right, let's go back to the beginning, and then we'll come back to the Krebs cycle. So I'll start off with glucose. What's the chemical formula for glucose? C6, H12, O6. Once I know that it's C6, I know the rest if I know it's a carbohydrate. Because all carbohydrates that are like single rings, they all have a ratio of CH2O, carbo, and then water is hydrate. 1 to 2 to 1, 1 to 2 to 1. Carbo, hydrate. So C6H12O6, so it's got 6 carbons, and these are my bonds, my electrons. These pins are inferior, probably fakes. So in glycolysis, I go along here and I basically break this in half. And when I do that, the reason it breaks in half is I'm stealing the electrons. What were these electrons doing before I stole them? Like, what was their job? They were bonds. So now, who stole these electrons in glycolysis? Yeah, the NADs. So that's my electron carrier. So NAD plus came in, grabbed a couple of electrons, stole them, and is going to use them to power these pumps. All right? Are there still some electrons to be had? Yeah, I can still steal these and these and these and these. So by the time I get to the end, I've got a pyruvate and another pyruvate. That's why I have 2 pyruvates. So I'm going to take this pyruvate, send it in here, and run it through the Krebs cycle. What's the whole point of the Krebs cycle? What do I actually want out of this? The electrons. So NADs and FADs are going to go in and steal all the electrons. I use those electrons to power pumps. I power this pump with a little bit of energy. I power the next pump with a little bit of energy. I power the last pump with a little bit of energy. All right, I've got these leftover carbons, so how do I get rid of them? I breathe them out as CO2. So my electrons go from pump to pump to pump. Now I've just got all these like waste carbon dioxides, and I just breathe them out. All right. When the electrons go from pump to pump to pump, they'll eventually just pile up on that last pump. But who was like my savior? A little Easter throwback right there. I thought that was pretty good. It's after school hours. I thought I'd be safe. Hardly anybody got that. Okay, so who's going to take the electrons away? Oxygen. So oxygen will come in. It's actually pretty good if you go back and watch that on tape. So oxygen will come in, grab electrons, and take them away. That's why I have to breathe oxygen. Because if I don't breathe oxygen, then the electrons just pile up on the end. I can't get rid of them, and I die. And I don't want to die. So I'd like to rise again. Oh, man, I'm on fire. Not on fire. It's like the opposite. Wish I knew how to edit video. All right. So that's cellular respiration. What is, try real quick on your paper, see if you can jot down the chemical equation for cellular respiration. And we'll end up closing with this. By the way, we always have like a bunch of stuff that's like on the table for the afternoon. Anytime we don't get to something, like last time, if we don't get to enzymes, if we don't get to photosynthesis, we do come back and like catch that like in class. So if you're like, hey, what about enzymes from last time? What about photosynthesis? No worries. So see what you can write down about that, and then we'll call it a day. Looks horrible. All right, I'll give you about 15, 20 more seconds, then we'll see what you got. All right, so on the reactant side, we got glucose, of course. What else is on the reactant side? Oxygen. Yeah, because I've got to breathe in respiration. In cellular respiration, I've got to breathe in oxygen. And then what do I breathe out? Yeah, CO2. And what else do I breathe out? Yeah, water vapor. So water. Six. I don't know what it is about today. And then what's the whole point of this? What am I trying to make a bunch of? ATP. And so textbooks will say 30 or more. Most high school books say 36, 38. Most college books say 30. It all depends on whether you're counting like gross ATP production or like net ATP production. So thanks for coming out. See you guys later. See you guys later.